So our next speaker is Dr. Tobias Bauer he's a postdoctoral researcher at University Hospital Bonn in Germany. Unfortunately, he couldn't be here with us today, but he was kind enough to send his pre-recorded talk, so we can all watch that together.
Hello, everyone. First of all, I would like to thank the organizers, Sarah, Boris, and Jonathan, for the kind invitation to speak here. And I must apologize for not being in Brisbane and giving this presentation in person. Before starting with the neuroscientific part, I wanted to start this with a little short overview over the hemispherotomy techniques or the hemispheric epilepsy surgery techniques, and an overview of what is actually cut in terms of connectivity. So just as a schematic, a healthy brain consists of two hemispheres, as we all know. They are connected, so they are interhemispheric connections. There is intrinsic connectivity in each hemisphere, and there is an input and an output to each hemisphere. This is actually a normal brain. And if we look at brains that have epilepsy on one hemisphere and epilepsy surgery, so a hemispheric epilepsy surgery is an ultima ratio option. There are three different techniques that I wanted to go through. So the first one being a callosotomy. In callosotomy, only the corpus callosum is cut, and this way both hemispheres are disconnected, which is aimed to prevent seizures to spreading from the lesioned hemisphere to the contralesional hemisphere. But still, both hemispheres remain with an intrinsic connectivity and have receive input and can generate output to the rest of the nervous system. Then a second type of surgery would be hemispherectomy or structural hemispherotomy. There, the lesioned hemisphere is removed entirely, so there's basically nothing left that one could analyze using MRI. But the remaining hemisphere still has its intrinsic connectivity and has input and output to the remainder of the central nervous system. And now the more interesting case that we are studying here is a so-called hemispherotomy or functional hemispherotomy, where the lesioned hemisphere is entirely disconnected from the rest of the nervous system. So it keeps its intrinsic connectivity, but it doesn't receive any input and doesn't generate any output to the remainder of the nervous system. And it's also disconnected from the other hemisphere. Why is this an interesting case to study? Basically, there has been a report or an opinion paper by Tim Bain and colleagues about islands of awareness, and they hypothesized that there could be three scenarios where isolated awareness may persist in disconnected cortical islands. This could be excranial brains and cerebral organoids, which is a bit more far-fetched than the actual case of hemispherotomy that we are studying here. And they hypothesized that awareness may persist in fully disconnected cortical islands, but they also acknowledge that detecting these islands of awareness may be difficult to do, but it can be addressed with neuroscientific techniques such as intrinsic resting state fMRI activity. What we did in our cohort that we had at our center and consists of 26 individuals who underwent hemispherotomy, and 9 of those 26 individuals had an isolated hemisphere that was anatomically sufficiently intact so that it could be used for resting state fMRI analysis. On the following slide, I wanted to guide you through the basically 4 steps of analysis that we did with that cohort, and that I want to talk about in the next 10 minutes or so. The first analysis being a very basic voxel-wise analysis, where we looked at patterns of activation and we're actually looking for a basic default mode network in those brains. The second step is where we constructed a network model using the correlation coefficients between pairs of nodes that then were ordered to networks. Third, we used a gradient-based approach to investigate the underlying hierarchy of networks and not using a network model from the outset. And the fourth and final approach that I wanted to show is the so-called brain fingerprinting approach, which is aimed to look at how brains, how the resting state pattern is specific to the similar brain and how it is different between individuals. So for the first analysis, the voxel-wise analysis, what we did is we extracted the default mode network from the healthy cohorts, from the healthy controls that we had, which are 24 individuals. And then we used those, the seeds that we got from the default mode network as seeds for the for the patients, 1 seed for the lesioned isolated hemisphere, and 1 seed for the contralateral connected hemisphere. And there we discovered, which is not too surprising, it has already been shown before, that in the connected hemispheres there is a default mode network which is then limited to this connected contralateral hemisphere. More interestingly, we also did this for the isolated hemispheres, for the 9 cases where we had the data and the hemispheres were sufficiently intact to analyze those hemispheres. And there also we found a pattern that resembled a default mode network. So taken together, there are basically the default mode network is at least the pattern can be found in both isolated and connected hemispheres, but it's not one network, it's 2 different networks, so 1 per hemisphere, unlike before surgery where this default mode network would be extended across both hemispheres. We had 2 cases which are even more interesting. Here we just looked at the post-surgical data, but we had 2 cases where we had a preoperative resting state MRI and 2 functional MRIs post-surgery. So there we looked into the individual networks. What we did here is to use the preoperative scan. This is an example of one of the cases, but you should trust me, the second case looks more or less similar. What we did is we extracted the default mode network seeds from the preoperative scan and then registered those seeds to the postoperative scans. And here we found that this exact default mode network pattern was still persistent, but then split across both hemispheres in the isolated and the connected hemispheres at the earlier time point, 1 month post-hemispherotomy, but also at the other time point, which was 5 months post-hemispherotomy. With that, I would like to move to the next analysis step that we did. Here we used the Schäfer atlas, which consists of 200 parcels, each of the parcels belonging to one of the 7 networks that range from the default mode and control network to unimodal networks like the somatomotor and the visual network. And then we constructed the network matrices based on the Pearson correlation coefficient. We did this for the control cohorts and also for our patients. And here we found that the connectivity for the difference between the connected hemisphere and the controls, we found that there was a higher connectivity in the connected hemispheres between the default mode network, the salience network, and somatomotor and visual networks. We also looked at the isolated hemispheres and compared them to controls. And here we found the same network connections that were increased in the isolated hemispheres of patients. But as a general tendency, mainly we found increase between network connectivity both in isolated and connected hemispheres between the transmodal networks, especially default mode network, but also control and limbic network, and the unimodal networks, so visual, somatomotor, dorsal, and salience networks. We looked at this also longitudinally. On the left side, you can see a plot for between network connectivity that we assessed in a mixed model, since we had several post-operative scans in the 2 cases, as I said earlier. And here we observed that between network connectivity in both hemispheres increased with time post-hemispherotomy. It increased even stronger in the isolated than in the connected hemispheres as a general tendency. And when we looked at the individual networks, we can see 2 things. The first being that this effect seems to be always stronger in the isolated hemispheres, which are shown in the darker lines or darker dots, than in the connected hemisphere with the lighter lines and dots. This is the one thing. And the second thing is that especially in the transmodal networks, between network connectivity, so the connectivity between default mode network and the other networks increases in particular, while this is less the case for unimodal networks. For the visual network, it decreases actually a little bit even. Then moving to the third step. So one problem with the approach that we have used for network connectivity is that the networks that we looked at, they are defined from the outset. So this is basically something that we put on the data from the top, and it's not really a data-driven approach. So we wanted to see if the 7 networks and the hierarchy from unimodal to transmodal networks, whether this is also preserved in the isolated and connected hemispheres. So we used a different approach, with a gradient-based approach, and is also based on the connectivity between 200 parcels, but we don't from the outset assign them to networks. So what we do instead is to calculate the correlation of connectivity between all parcels, and then use a diffusion embedding approach to define 2 gradients. So two gradients of organizational axes of the hemispheres, and then assign the parcels again to the 7 macroscale networks. If we color them according to the macroscale networks that they correspond, we find that the first gradient ranges from unimodal networks to transmodal, or to the default mode network area. Here the first gradient in the controls is colored from white from unimodal to the red colors for the default mode network. Whereas the second organizational axis, or the second gradient, in controls ranges from the somatomotor to the visual network. When we did the gradients also for connected and isolated hemispheres, basically what we can observe is that the pattern itself looks very similar to the control hemispheres, for the connected hemispheres, but also for the isolated hemispheres. So now let's move to brain fingerprinting as the final approach. For brain fingerprinting, we split the bold time series into a first and a second half of the resting state fMRI scan, and then we calculated separate connectomes from the first and from the second half of the resting state fMRI scan. And the fingerprint basically is the correlation between the connectome of the first and the connectome of the second half of the MRI scan. And we did this for all the controls, all the patients, and then what we get is a so-called identifiability matrix, for which the x-axis are the first half of the subjects, and the y-axis are the second half of the subjects, which means that the diagonal elements are the self-identifiability, so how similar the first to the second half of a specific subject is, and the off-diagonal elements is the other identifiability, so how similar the first half of one subject is to the second half of a different subject within the same group. We found that the self-identifiability didn't differ between patients and controls, not for the isolated and also not for the connected hemispheres. But when the right hemisphere was isolated, we found that the other identifiability was lower in patients than in controls, which means that the first half of a connectome in patients in the isolated hemisphere, if it was the right hemisphere, was lower than the correlation to other subjects within the group than it was to the subject itself. The other way around, when we looked at those brains where the left hemisphere was isolated and the right hemisphere was the connected hemisphere, again the self-identifiability, so the correlation between first and second half of the same subject, wasn't different between patients and controls. But again, for the right hemisphere, the other identifiability was lower in patients than in controls, if the right hemisphere was now the connected hemisphere, which is a bit surprising because it's always the right hemisphere that seems to have a lower other identifiability, regardless of whether it is the connected hemisphere or the isolated hemisphere. Basically, those were the 4 steps of analysis that we did with our cohort so far. And I hope I could provide some of the insights to how isolated and connected hemispheres of the brain are organized after hemispherotomy. And to sum this up, on the voxel-wise analysis, we found that the default mode network is per se intact, but it's split between both hemispheres. In terms of network connectivity, we observed that between network connectivity increases after hemispherotomy. So it shows an increased integration of unimodal and transmodal regions. The gradient-based hierarchy approach showed that in general the hierarchy organizing the brain or the hemispheres from a transmodal to unimodal networks is overall preserved. And the brain fingerprinting showed that the self-identifiability is also preserved in isolated and connected hemispheres after surgery, but we also observed a higher idiosyncrasy of right hemispheres, regardless of whether they are the connected or isolated hemispheres. And by that, I would like to thank the organizers again. I would like to thank the team behind all this research, and I thank you very much for your attention.